Delaney. Thank you, Kurt. I forgot to mention to Kurt that it is being recorded. Dr. Delaney is going to be talking about what we know about honeybee diversity. She comes from that with a great deal of experience in looking at bees and determining what their origins and parentages may be. We sent out a bio for each of our speakers, so I'm not going to repeat that, but I will add to Dr. Delaney that she has a WAS connection. She was here at Oregon State for her master's degree, was a commercial beekeeper for a while in the state of Oregon. I went to Washington State University and finished her PhD at Washington State University and taught for a while during her degree program. She has been now at the University of Delaware since 2010, I think you started, roughly 2010. She's an associate professor there, and as you see from her bio, she is extremely active with teaching and a very active extension program, as well as doing some very significant research. So this evening, she's going to start off. She's going to be talking about what we know about honeybee diversity. So Dr. Delaney, the floor is yours, and welcome to our WAS April mini conference.
Well, thank you so much for inviting me, and that was too nice, as always, Dewey. Yeah, my role is definitely more shifted towards teaching now, so I just kind of do research that is interesting to me instead of feeling like I constantly have to do research. But let me share my screen with you all and find where my talk is on my desktop. Here we go, and get it started. I will say that I very much always miss the Pacific Northwest, specifically Oregon. And me and Dewey kind of switched, we traded spaces. But yeah, so I'm going to talk to you about understanding honeybee diversity. And I present it kind of like I do understand it and I don't. So it's something that I'm curious about. And so let me just say I'm trying to understand honeybee diversity, and I will share with you my thoughts and kind of how I interpret some of my data. Let me move forward here with the slides. And so I just wanted to show this picture because I love it. This is a beautiful stigma with pollen grains on it. And I like it because this really horticulturally, scientific in a nerdy kind of way embodies pollination, right? Or after pollinations, because pollination is just the movement of the pollen, the male gamete to the stigma. But this is kind of encapsulates why we care so much about honeybees, not just honeybees, but other insects like bumblebees and monarchs, all these things that help to move pollen from anther to stigma. And another thing that we think about when we think about pollination, especially to this group that I'm talking to, is beekeeping, and the fact that many of our beekeepers in this country will travel their bees to different crops specifically so that their bees will move pollen to stigmas, right? And then that will lead to a good yield. So this is one I just pulled off the line from Dave Hackenberg. I don't even remember what year this is from. But a lot of people, unless you're a beekeeper, don't really understand the journey and what pollination, commercial pollination entails. And how important it is until you hear of an accident, right? On the freeway, and you're like, why are bees on semi trucks, right? And another thing that we have to deal with is the fact that I would say earlier than this, but 2006 is really when it hit the news. We've seen colony failures annually of about 30 to 50%, give or take a years, right? But, these reports come back every year. Bee Informed Partnership does these. Other people are reporting. There's a lot of surveys out there. And we have seen these kind of catastrophic losses coming out of winter a lot of times. And so this has been a headline in the news. And because of this, tons of research dollars have been given to land-grant universities and been kind of doled out from various organizations to kind of figure out what's going on here. So we've looked at everything in honeybees, viral loads, morphology, beekeeping practices. And the more and more we look, the more and more we learn. But I still don't think we've found a wonderful solution to some of the issues. And one thing I can tell you that I, as well as you, because I was a beekeeper before I was a bee researcher, do is we love the biology of the colony. We understand that when we look at our bees, we're not just looking at the health of an individual, the health of a queen per se. We're looking at the health of the colony. And in order to do that properly, you really have to understand the biology of the colony. And that goes all the way back to how they live. And if we think about honeybees, they're part of this unique group of insects that exhibit eusocial behavior or true sociality. There's not many insects that actually are considered truly social. Termites being one of them, all ants, and some bees and wasps. And why am I bringing this up and why do we care about this? Well, it's because when we think about a honeybee colony or an ant colony or a termite colony for that matter, you're dealing with these very large family groups. And because you're dealing with large family groups, you're dealing with different types of relatedness. You're dealing with organisms that are living close to one another, that are living close to their food. They're living close to their young. So there's all these different crazy variables going on with these large family dynamics. Eusocial insects, by definition, have an overlap of generations. So we have one generation taking care of the next, etc. Right? Also, there's a reproductive division of labor. Not all individuals reproduce. Hence, we have these queens that are so important to colony functioning. We also have cooperative rearing of young, where we have a generation of sterile workers that are helping to raise and feed the next generation. Some other really cool things that we see with eusocial insects, or truly social insects, is this ritual feeding, this trophallaxis behavior. And also, generally, eusocial insects have complex ways of communicating using pheromones and chemicals. And we know with honeybees, there's a whole bunch of them. Probably one that a lot of people are familiar with are alarm pheromones, but there's a lot of really interesting ones associated with the queen as well. And also their nests, and I think this is one of the coolest things about eusocial insects, is often temperature controlled, humidity controlled. I mean, we think honeybees are really amazing with the way that they can maintain a very static and internal environment in their nest. Look at some of these giant termites and your head will blow off your body. That's so amazing. And this is the one that always raises the hair on my neck, is that eusocial insects, they have specialized reproductive biology. They don't just produce new individuals, they produce new colonies. So they swarm and give birth to a new colony. And it's kind of this colonial birth, right? Or offspring that they're putting out into the world. So because of all these amazing things and because of their genetics and their relatedness, what we see is this sterile caste. And this sterile caste of workers is really something that threw even the greats like Darwin off, right? They were like, why would an organism like not reproduce and actually raise other individuals' offspring, right? And so that was something that really plagued a lot of early naturalists. Some individuals reduced their own lifetime fitness or reproductive potential to raise the offspring of others. And we now have learned since then that actually that's really important to the functioning of these large family groups, how we can understand the health of these large families that live so closely together with each other and their food. And their relatedness is something that's very, very important. And this is going to get at this idea of diversity. All right, so how can we kind of overall look and understand colony health? Well, just as we're beekeepers, right? We go through, we look at them, we can smell it, we can look at the way they're moving their bodies, the way they're behaving in the hive. A lot of us, when we've worked bees for a long time and we have lots of beehives that we've gone through, we have different go-tos of things that we essentially look at first, to understand the health, and then we can kind of fine tune when we see things that aren't quite right looking in the colony. We go to another kind of detective sleuthing level within our colonies. We don't do this just as beekeepers, but as researchers, we do this too, in a much more calculated way, where we'll actually look and go frame by frame and maybe take notes and kind of put quantitative numbers to the things that we're seeing so we can run statistical analyses on them. Well, one thing that I've always been intrigued with, and also, yes, understanding honeybee health for sure, but also understanding the genetic diversity of bees in the United States. Honeybees pose a very interesting scenario. And this paper put out by Carpenter and Harper in 2021 is really great. If you have not read it, it's in Apidology, definitely check it out because it's really well done. And it looks at the genetic past, present, and future of honeybees in the United States, which is something that I am super into. And you can see the dates here. You can kind of see these different subspecies names here. And these are basically showing introduction events that we know of these different subspecies into North America. And so understanding the genetic past of these bees in North America, because they are not native, they were brought here, is super important and informs us about the honeybees that we have here and how they're interacting. So we're going to look at honeybees from many different scales of diversity. So the colony level, we're just going to touch on that a little bit. We're also going to look at it at the population level. And even more kind of in a larger kind of context as well, looking at many different data sets. But one way that I have learned to look at honeybee populations and honeybee colonies in terms of diversity is through the use of molecular tools, specifically mitochondrial DNA and nuclear DNA. And we've advanced much more than when I was a student doing this in the lab. Now I kind of tell people what to do, or they're telling me like, oh, Dr. Delaney, this is a much better assay to look at this. And I'm like, how do you do it? Because things are advancing so fast. But overall, we can still kind of focus on these two different types of DNA. Mitochondrial really is focusing on maternal ancestry, as well as other things, but we're going to talk about maternal ancestry here, as well as nuclear DNA, which is really useful for the population kind of work because you're getting both genetic information that is passed on from the males in the colony as well as the females. So you're getting a different kind of picture. You're tracking the contribution from drones. And this is just a diagram kind of showing this. So this is through the mitochondrial DNA. So you can see that basically when you look at each generation, you are looking at maternal ancestry, right? So the mitochondrial DNA will reveal kind of the past ancestry on the mother's side, on the queen's side. Whereas nuclear DNA, you're going to get information looking at this queen from both the queens of the past and the males of the past. All right, so another thing that makes honeybees and eusocial organisms in general just kind of complicated, especially in the order Hymenoptera, where we have our bees, ants, and wasps, is that they're haplodiploid. And really all that means is that females are diploid, meaning they have two sets of chromosomes, and males are haploid, they have one set. And this haplodiploid nature is not only a sex determinant, so males are haploid, females are diploid, but it also has repercussions on relatedness and when we combine it with kind of the sexual behavior of honeybees. Oh, goodness. So some work that I have been involved with looks at polyandry. And so this is this idea, we know that honeybee queens are highly promiscuous and they mate with multiple males. And we've seen, and many papers will say that Apis mellifera queens, the average mating is between 12 and 20 males. This can vary depending on the subspecies, but here in the US, most papers are going to showcase about 12 to 20 drones that the queen has mated with. And we've been able to even kind of test this and see if we can get artificially inseminate queens with even more drones. So kind of really increasing diversity at the colony level. But let's just look at this natural kind of diversity, this natural polyandry. And polyandry, this idea that this queen mating with these 12 to 20 drones is getting genetic contributions from these 12 to 20 males and holding that in her spermatheca. And that when she gets back to the colony and she starts to lay eggs, when she actually fertilizes that egg, creating a diploid female, that diploid female is actually going to have different relatedness values with some of the other diploid females. And so some of them are going to be super sisters, meaning they share genes from their mother and the same father. And some of them are going to be half siblings, where they're going to share, of course, genetic information from their mother, but they're going to have different fathers. So if we take a sample of workers from a colony and we look at their genetics, we can actually determine the number of patrilines or subfamilies, or how many drone daddies were, the queen mated with in order to make these worker offspring. So this really is great because it gives us an idea of colony level diversity. And if we kind of look at this, this relatedness coefficient, and this goes back to what I was talking about, where altruism, where you're giving up your own reproductive fitness to raise the offspring of others. And long ago, this kind of kin selection idea was based on if you were more related, then you were more likely to be altruistic. So this really is showcased beautifully with honeybees, because if we look at diploid organisms like ourselves, full siblings are about 0.5 related, this relatedness coefficient. If we look at haplodiploid organisms, half siblings, or the relatedness coefficient is 0.25. Now, and so this particular worker, the sister here, they're half siblings because they share the same mom, but they share different dads. So their relatedness coefficient is 0.25. But this is the kicker. And this is why these workers will forego reproducing themselves and raise the offspring of this queen, because full siblings, super sisters that share the same dad, they share 0.75 relatedness coefficient. So you can see that it's in their best interest to raise the offspring, hopefully the super sisters, of this queen mother, because it's making more individuals that are like them. And eventually they're going to make another queen that's going to have genes that are similar to theirs. So our queen stock industry is founded on the identification of colonies, of course, with desirable traits. And that's going to be different depending on the needs of the industry, the needs of the beekeeper who is purchasing it, and so on. And this basically is done by selecting queen and drone mothers with these desirable traits for the next generation. And it is a double-edged sword, because we know we want diversity. Right? We know diversity is good. I'm just going to show you studies that showcase that in a minute. But we also want to select for these desirable traits. And we've seen this a lot of times in different studies and different lines that have been developed throughout the years, different stocks, races, where we have these special traits that have been selected for hygienic behavior, VSH. I always want to make sure I'm not saying VHS. That's a tricky one for me. Varroa sensitive hygiene, VSH. But even like the Russian bees as well, when they first were brought over to Baton Rouge and quarantined, and working with some of those Russian lines. Because honeybees, we can control the mating on both sides, but the part that's tricky is the fact that there's just so much diversity built into the behavior, built into honeybees, that it's really hard to keep drilling down and selecting for desirable traits without losing, one, other traits that might be useful, and two, keeping these bees diverse enough. So benefits of diversity, like I said, diversity is built into honeybee biology. Everything about a honeybee is meant for diversity. The fact that the queen is promiscuous, right? The fact that they're haplodiploid, the fact that they swarm and move quickly across the landscape. I mean, if you don't manage your bees for swarming, you can see how quickly these swarms will move, and your bees will swarm, right? And there's so many different studies. This is just a few. Here are some of the citations down here where these studies showcase the importance of diversity at the colony level in terms of resistance to disease, both chalk brood and American foul brood, to a diversified workforce. That is the benefit of diversity, that some subfamilies, so these matrilines or patrolines, perform different tasks more frequently and maybe better than others, right? Foraging, thermoregulation, cleaning, hygienic behavior, and not just natural grooming. So that has been shown to that diversity promotes these different types of behaviors and a diversity within these behaviors. Benefits of diversity, the study by Madeline Sealy, swarms found colonies faster, or like locations for them to settle down, right? Higher weight gain, more food stores, an increased foraging rate, better scouting by foragers, more beneficial microbiota in the gut even. So, colony levels that are sired or headed by a queen that has multiple mates compared to one that is just mated with one drone has better gut microbes. That's just amazing. That is amazing to me. So how diverse are our bees? We can see it's been shown, it's been proven that diversity at the colony level has many, many benefits. But how diverse are our bees? And this is where it gets kind of tricky, and I will tell you why. If we look at the species Apis mellifera, there's many subspecies. These are based on morphometrics and have been supported by molecular analyses, that there's over 24 recognized subspecies. So there's a lot of support behind this subspecies designation, to much dismay by many taxonomists, because many taxonomists don't like subspecies. But here is a list of some different subspecies and the corresponding location based on color. And if we kind of look at honeybees in their native range, and we look at all these different subspecies, they cover a vast, vast area, and not just a vast, vast area. And the ones that you're not really seeing here are a lot of the Africanized subspecies or African subspecies. But so even a larger area or natural distribution of this species. And there's so many different climates, ecologies. And so you would expect that just like neighborhoods, these different climates, these different ecologies, different times of flowering, different amounts of flowers, they're going to shape the behavior. They're going to shape the morphology of these bees. And that's how we can identify these different subspecies, by these kind of evolutionary pressures forcing and acting on morphology, on behavior, because they have to deal with these different types of environments. So this is again from that Carpenter and Harper paper. And again, just showing you the spread of Apis mellifera when kind of this European settlers kind of came to North America, bringing bees with them, and what this spread looked like. And so I definitely recommend you looking at this paper. And what I want you to get from this is that you just saw the native range. You saw how diverse, just not only in kind of the range, in subspecies, but now let's think about this. People brought a few of those subspecies on a ship to North America. And basically, it created an initial founder population or a bottleneck to the diversity of or within Apis mellifera. This is from a paper by Steve Shepard in the late 80s. However, regardless of importation, the fact remains that less than 1/3 of geographic races of Apis mellifera present in the old world have ever been transported to this country. And this is not just honeybees. There's many different organisms where we've created this founder population, where we brought something from somewhere else. We just took a little snippet of the diversity and then started a whole new population in another part of the world. This is my feeble attempt to create a visual of a founder population. So this is the initial population, very diverse. You just take a snippet of that diversity, bring it across, and then you have this less diverse population. And this is all you got. There's no other inputs coming in. So there's this limitation at the get-go in terms of diversity. Another bottleneck that has been seen is just in the way that we breed bees. And we breed bees pretty amazingly well here in North America. I've seen these operations, they're fantastic. But even when we think about how many queens we are creating, there's still only so many queen mothers that these different breeders can use in order to generate the demand and the need for new queens to be mated and sold and given to other beekeepers. So in different years where we did this sampling, we saw 603 queen mothers used to produce close to 1 million and 473 used to produce close to 1 million. And so these has a potential to further reduce diversity. So if we look, and we've looked at the genetics of US populations, kind of what the Carpenter and Harper paper looked at, but specifically trying to understand, okay, what do we see now in our honeybees in the US? And we kind of broke them into Western and Southeastern commercial breeding populations. And this work was done in the 90s and then redone in the early to mid 2000s. So this already could probably be done again, though honestly, looking as long as I've looked at the genetics of bees, I don't think we'd see too much change. But one key point that came out of this work was that the Western commercial breeding population here in the green and the Southern commercial breeding population here in the red were genetically different from each other, not only in the early 90s, but that still continued into 10 years later into the early 2000s. And I would say if we were to do this study again now, we probably still would see that separation or that they were genetically different from one another. And this not only is due to the fact that we had different genetics being introduced to the West Coast early on than the East Coast, but also the fact that they were kind of geographically isolated for a very long time when breeders were starting to breed bees. So there's a lot of different factors that have kept these two different areas genetically different from one another. If we look at mitochondrial DNA, so genetic diversity from the mama, the main thing I want you to see here is in that same kind of commercial stock, looking at commercial queen breeders from the early 90s and the early 2000s, most of the maternal ancestry of these bees are C lineage. So this would be Ligustica and Carnica. I mean, there's some other things in there, but really the big take home is Ligustica and Carnica, right? So if we also look at the populations of US honeybees and we look at feral populations, so these are colonies that are not maintained by beekeepers. So they aren't necessarily, feral is a loaded word. Let me just put it that way, because we don't know if they're swarms from, that have escaped from managed colonies. We just know that they're not at that point being maintained by a beekeeper. So it makes it kind of tricky. I can tell you that populations collected between 1980 and 1991 by Steve Shepard and Nathan Schiff were definitely more of what we considered truly feral. And when we looked at the DNA of about 700 feral colonies, we could see here, again, this large maternal signature of Carnica and Ligustica, but a large portion of this genetic pie was Apis mellifera mellifera subspecies, representing different variations or haplotypes within Apis mellifera mellifera. So this is something that's very unique and characteristic of this old kind of feral population before Varroa. So that's pretty interesting and unique. Another thing is when we compared commercial populations to these feral populations that were collected near that time, we found that the commercial and feral populations did have a different genetic structure from one another. So there's been a lot of ideas that there's been this loss of the feral population, and there's been many studies. I can just speak to mostly to the one that I was involved with, where we wanted to understand what was making up these unmanaged bees. We called it the Feral Bee Project, but really we were just collecting bees from unmanaged colonies or hives. So it could have been in a tree, in a church, wherever. And then I would bring them back to the lab and look at them to see if there were any genetic remnants that kind of looked like those old feral populations collected in the 80s. So one tool that we threw at these unmanaged bees was morphometrics. Morphometrics is painstaking, laborious, but it's really great at kind of understanding. It's a powerful tool for understanding subspecies. And this is where you will get a sample, you line them up, and then you basically quantify different morphological features. And you can run statistics on these and get different subspecies based on tongue length, the coloration on the abdomen, the width of the abdomen. I mean, there's so many different features. One feature that's really useful are wing angles. So angles and venation in the wings themselves, which are highly kind of pressured through evolution. So if we look at these unmanaged bees that we collected, and this was probably close to 300, 400 unmanaged bee nests. You can see here this green circle are unmanaged and managed bees that we collected from North Carolina. These are actually true species types of Apis mellifera mellifera and Apis mellifera causica, true subspecies types of Apis mellifera ligustica and Apis mellifera carnica. So what's really interesting is that mellifera and caucasica, based on these wing angles, morphometrics, kind of grouped together, and Carnica and Ligustica grouped together. But the North Carolina managed and unmanaged bees grouped together and didn't group morphometrically with either of the subspecies or any of the other subspecies that they probably originally were derived from at some point. So I always say this is like showing you that we're creating this kind of US mutt, right? Or something of that nature, at least based on the morphometrics of these wing angles. So when we were doing this study looking at bees and their genetics in North Carolina, specifically these unmanaged bees, we came across a lot of nests that were in water meters. We called them the water meter bees because we found so many of them. And also just these small kind of colonies that wouldn't stay in place for very long. And so their overall we were getting concerns. People were concerned that there was low levels of Africanization in the Raleigh area. So this idea that people were seeing small usurpation swarms, higher stinging rates, odd physical characteristics, strange nest locations. And so one thing that I decided to do with one of my first students at the University of Delaware is to take these unmanaged bees and compare them to some other bee sample sets. So unmanaged bees along the East Coast, at that point, I think we had close to 600 different colonies that we sampled that were just, not being managed, found in old structures. And then we had managed bees that were collected in North Carolina, Tennessee, and Delaware from known colonies that were managed. And then we had known Africanized colonies that we were looking at as well. These were identified as Africanized by the USDA ID. So this is, again, using morphological features or morphometrics by the government in order to determine that they were Africanized. So these were known to be from Florida, Alabama, Georgia, Arizona, Brazil, and Belize. And then we had known African samples from Kenya and Pretoria. And we decided to look at all of these samples with different tools to see what the different tools said about these samples and how they related to one another. So the first thing, this morphometrics, this geometric wing analysis, and this is kind of what this would look like. If you would put the wing into this program, it numbers each one of these wing angles and the intersection with a different wing venation. And this is how it makes measurements in order and kind of determines how to group these different types of bees. And this is the morphometric analysis of this Africanized sample set. And we had Dr. David de Jong from Brazil do this for us. And you can see in the kind of blue circle was Alabama, the red square was Florida, and the green, I think it's a diamond, was Georgia. So these were Africanized samples that were ID'd using the USDA ID. And then we had the Arizona. So this is an Africanized population from Arizona. And then let me go back one, sorry. And then we had the Brazilian samples here. So you can see how they separated out. They separated out differently from one another, but each one of the groups kind of grouped together based on their wing angles. Now, looking at molecular analyses of these different sample sources, what's really interesting is when we looked at the mitochondrial DNA, so the maternal ancestry of the Alabama, Georgia, and Florida samples, the Arizona samples, the African and Brazilian samples, and the Belize samples, all of these here can be considered non-Africanized. All the ones down here, these are maternal ancestry that are associated with African lineage. And you can see the Alabama, Georgia, and Florida, there's a few just that had an African lineage, but many of them maternally had European maternal ancestry. Now, if we look at the Arizona samples again, you can see here, obviously, these were mostly had African ancestry, African Brazil samples, African ancestry. And the Belize, again, was kind of a mixture where there was just a few that were European, but most of them had African maternal ancestry. So now we're getting a different kind of picture looking at the maternal ancestry of these same samples. Now, this is a genetic structure analysis on these same samples, but now we're looking at nuclear DNA. So we're looking at DNA contributions from not only the mother, but also the drones. And the main thing I want you to take away from this is you can see the African and Africanized samples from Belize. The African samples are here, almost purely yellow. The Africanized samples from Belize are here. The Africanized samples from Arizona. Then the ones that were collected from Florida, Alabama, and Georgia, we're seeing some integration, right, from Africanized bees, which is kind of represented by this yellow. But these managed bees, we see some spikes, but overall the unmanaged and the managed, it's mostly kind of this blue genetic mixture that is not associated with these more Africanized bees and African bees. So we're getting three kind of different pictures of these same samples based on the different tools we're using. So that's why I say it's interesting, but I still don't understand any of it. And if we look at the diversity of Africanized bees, too, like if we look at allelic richness, which has to do with kind of the number of genes, we'll just kind of simplify it that way. Private alleles essentially is alleles that are completely unique to that group, right? And you can see that the Africanized, at least the ones from Arizona, Belize, and from Africa, have higher levels of these private alleles that are unique only to that particular group. All right, so, and kind of trying to understand, those are all like way bigger pictures of diversity. So we went to the colony, then we went really far out. Now we're going to look at a population. And many of you are probably familiar with this work. I don't know anybody who is not, because Tom is just so amazing at telling the story. But I helped or worked with him to look at the genetics of the bees in the Arnott Forest and to investigate their genetic structure, because he has been mapping them, these tree bees. It's from the 70s to the early 2000s, and again in 2012. And in 2012, again, he found 10 bee trees in this Arnott Forest. You can see it's a 16.5 kilometer squared forest that is further surrounded by forests, right? So this is a pretty large tract of forested area. And he was able to give me bees from those 10 trees. And then he scouted around the Arnott Forest, and the only apiaries he found, one was 0.5 miles away and one was 2.5 miles away. So he sampled all the colonies from those, and he sent those to me as well. Because what he was really interested in understanding was whether these Arnott Forest bees were genetically different from these managed bees, any managed bees that were near them, right? So if they were unique, if these bees are kind of like just persisting in this forest, they're just not as constant escape swarms from managed bees. So we set out to look at their genetics. And again, this is the tree bees here. This is that apiary 1, 0.5 miles away, and apiary 2, I forget, but not too far away either. But allelic richness is the overall amount of alleles, or kind of a proxy for genetic diversity. The higher, the more diverse. Private alleles, again, are kind of genes that are unique to that particular group. And you can see that the tree bees are much higher in terms of their private alleles. So this is the first kind of idea that these bees are different. And so that's pretty darn interesting. If we look at the levels of genetic differences, and this is just based on these allele calls that we make looking at these different bees, we can kind of get a different genetic structure that's making up these different, the tree bees versus apiary 1 bee and apiary 2 bees. And what we can see is that the Arnott Forest bees are genetically different from the bees in apiary 1 and the bees in apiary 2. So this is interesting because these bees have their own genetic signature, right? So kind of leading more into this, we wanted to understand, okay, there's been concerns that maybe our bees aren't mating enough, commercial bees, and maybe that's leading to a decrease in genetic variation, which is affecting the colony functioning. And so we decided to look at the mating frequencies of honeybee queens in these feral bee trees in the Arnott Forest. Not kind of understanding what we might find, but overall, I was like, I bet they mate more. I bet they have more mates because they're feral, and they probably want to increase the diversity, etc. Well, we were surprised because what we actually found is that there was statistically absolutely no difference between the amount of mating with the R0 forest bees and the bees in apiary 1 and the bees in apiary 2. Just goes to show you that queen breeders are doing a pretty good job of providing diversity for their queens to mate with, that there's absolutely no significant difference in mating frequency between managed and unmanaged bees. All right, so the last thing I'm going to talk to you about is a paper that we just got published this year. And this is work that we had collected quite a while ago, and honestly had sat on for a while because we just weren't sure the right time to put it out. But I think this is just so interesting. And this is the meta-analysis of genetic diversity and intercolony relatedness. And this is now looking at how related reproductives are in commercial honeybee populations. So are our queens mating with brothers? Right? That makes for a sexy title, right? So we were aiming to look at these sample sets and kind of quantify the degrees of relatedness at the colony and also at the population level and operation level in some cases. So I just want to first introduce you to the data sets. You guys have been a fabulous group. Really, I kind of have you in prison here. You have to listen to me, but I'm almost done is my point. And so the sample sets we have are an Africanized honeybee sample set. So from 17 different colonies in Arizona, in rural Arizona. We have 79 colonies from a migratory operation. We have 22 colonies representing commercial breeders, and we have 61 colonies representing commercial breeders from the following year. And then we have 33 colonies representing experimental colonies where we actually used super sisters or sister queens. So this basically is a highly related sample group. And so what we wanted to do was we wanted to look at the relationships between queens in these Africanized honeybee populations, between drones in these Africanized honeybee populations, between queens and drones in these honeybee, and look at those kind of relationships between the reproductives in each of these sample sets, and then compare them to one another. So that's what I'm going to try and walk you through. These are the different microsatellite loci we looked at on the nuclear DNA. And this is just a visualization kind of looking at the comparisons we made from population to population. We looked at queen to queen here, queen to mate. We looked at mate to mate and drone to drone. So let me walk you through some of this. This is the average genetic relatedness. And you can see here, our populations are down here on the bottom. We have our Africanized honeybees here from Arizona, our migratory group from all along the eastern seaboard. We have our commercial population group 1, our commercial population group 2, and our experimental group where we were using sister queens. And you can look, this is drone to drone. This is queen to queen within these groups, mate to mate within these groups. And then finally, or up here, we have queen to mate up here. All right. So the difference between mate to mate and drone to drone, this is drone to other drones in the population. This is drone to other drone that mated with the queen. Okay, so that's kind of the distinction there. And the big take homes from this, because it's really interesting. Overall, you can see that this kind of serves as our group that represents true genetic differentiation, right? They are not related to one another. They're very outbred. Whereas this experimental group where we're using sister queens, this kind of represents our inbred group, where we're closely, there's a lot of relatedness going on. We knew that going in. So what we can see is in all these groups here, these letters designate whether they're statistically different from one another. These intermediate groups here, these other groups are kind of intermediate and statistically different in certain circumstances. The first one I want to point out here is this one, where you have the three commercial populations were kind of numerically intermediate and all above 0. Right? With the breeding 1 population, so this is the commercial population, being statistically equivalent to the experimental population, which is kind of the inbred population, and the migratory population being significantly lower, and the breeding 2 population being between the migratory and the AHB populations. So that's just kind of interesting, kind of looking at how the queens are, the average genetic relatedness of queens in these different groups. Another take home is looking at these drones. The experimental population in the drone relatedness was statistically lower than that of the commercial populations. So the ones where we were kind of controlling the mating, we had sister queens, the drone relatedness was lower. They were less related than that of commercial populations, suggesting experimental research apiary had a higher genetic diversity of drones than tested commercial populations in this snapshot of samples. Remember, so that's one thing you always have to keep in mind with all studies. This is just a snapshot based on the samples that we had access to. Now, this next chart is really interesting because it's looking at the average kind of relatedness within these different populations, again, within these groups. So the dark black represents, in this one, the mate to mate, brothers, the gray is cousins, and the white is unrelated. So you can see the amount of brothers in the AHB, the amount of brothers in migratory, and compare them across the amount of cousins, the amount of unrelated. And so this would be drones. Again, we have brothers across the different groups, cousins across the different groups, and then unrelated. You can see queen to mate. Again, there's no real differences at all, but now we have these siblings in the black, cousins and unrelated. The thing that I want you to take note of is this queen to queen is where we're starting to see significant differences in the makeup. And what we see here is that in the kind of slashed here, this is the amount of super sisters, right? So experimental, we would expect a higher amount of super sisters because we knew that was part of the experimental setup. And then if we look for AHB and experimental populations, we're minimally and maximally inbred, right? So the AHB, we know they are very much outbred, right? So we don't expect to see a whole lot of highly related individuals there. And the 3 commercial populations were all intermediate, each having more than 48% of the queens being genetic equivalent of super sisters or half sisters or cousins. So it's just interesting. I don't think there's any big profound things that I can say, like big generalizations, except that it depends on what you're looking at, right? Diversity is interesting, and it depends on what you're looking at and what level you're looking at your bees. I think it's an interesting idea to look within a population to see how related our mates are. And it's hopefully this can be something that can be used as a tool in the future to better understand the health of our mating populations. This is, again, these same populations, but now looking at nuclear DNA, kind of looking at the population distinctions in terms of their genetic makeup. You can see the AHB here is very genetically unique. We can see migratory. 1 here is kind of a mixture, an admixture, this breeding 1 here, breeding 2 again, and we have this experimental where we've really kind of narrowed down the genetics. And so we have these 5 different kind of groupings here. So pretty interesting looking at these different populations in these different ways using genetic tools. Key takeaways from this: commercial breeding populations of managed honeybees are more inbred than maximally outcrossed, but they have sufficient and significant genetic variation depending on the level of analysis. And a lot of people, and me included, were really kind of on a witch hunt to look at and say, oh, there's just not enough diversity. But one thing I've noticed with honeybees in the United States is that they are very diverse, but they all have the same high diversity, making them not diverse from one another because they all have the same high level of diversity. If that makes any sense, probably not. But if you look at it on paper, it's the same with many introduced species. Alfalfa leafcutter bees are the same way. There's a lot of variation, but all of them have this variation, so making them not that different from one another. So this leads to the fact that there's minimal structure within commercial honeybees because there's a lot of diversity, but they all have it. So you don't see one group really standing out uniquely from another group, right? So this highlights that the population in the US is completely panmictic. Is not completely panmictic, but nearly so. Very, very well mixed, so to speak. So another key takeaway is that really to understand the health and how it's tied to diversity, it depends on the scale in which you want to look for that diversity. All right, so I know that was a lot, and hopefully I explained some of it well, but I don't know if I'll probably have time for questions later. I'll let Dewey take over, and thank you for listening, and I'm excited to hear Carolyn. Let me stop sharing.
Thank you, Debbie. Very interesting. We are going to take a break. There are a couple of questions that came in, and if we have some time later, we can try to handle some questions. We're going to take a short break, and then we'll start with our second presentation, Carolyn Brees from Oregon State, and we'll give you about 4 or 5 minutes to get off to the bathroom or get your snack and then get back. Paper, that reference, Tarpey Carey and her own publication, Frontiers of Insect Science. This is available. So if you want to capture that to look at the aspect, some of the questions were very specific questions related to definitions within that study. So that's why I wanted to have that brought back. All righty. Thank you, Deb. Appreciate your information. A lot of nice comments that are appearing about your talk. Appreciate it very much. We're going to switch gears for our second talk. Our second talk this evening is going to be by Carolyn Brees. Carolyn is going to be talking about engaging beekeepers with exciting programming. Carolyn got her biology degree at Oregon State University, went off to Arizona and got a degree in forestry, her master's in forestry. And then coming back to Corvallis, Oregon State, has been our manager for our bee apiary colonies and taking care of the colony since 2009. She's also teaching. She does a very progressive online course at Oregon State University about bees and apiculture. And I liked her comment in terms of her bio. She says that her hobbies include keeping bees, talking about bees, worrying about bees, and dreaming about bees. So she's like most of us, I think. So Carolyn, welcome. Thank you for joining us this evening. Carolyn's going to talk about engaging beekeepers with exciting programming. Carolyn, it's all yours.
Thank you. Thanks, everyone. Thanks for the opportunity to speak at the Western Apiculture Society. It's a real honor. And let's talk bees. So tonight, I want to share with you some of the educational programs that we have at OSU that get people learning and building community. Let me reduce this here. Okay, so a little bit about me. Okay, so I started at OSU in 2009, and I helped develop the Oregon Master Beekeeper program and coordinated it until 2017. At that point, I had the opportunity to move back into bees and do more research, and I still do some extension. So I'm excited to tell you today about some of the things that we've done in the past and that we're still doing in the future, or doing now, and that we'll do in the future. And from what I understand about you is that you are the movers and shakers of bee clubs. You're always looking for ways to engage others, and you, as the WAS group, you're obsessed with bees, and I know this because it's a Tuesday night and you're here listening to bees, about bees and science and programs like this. So it's an obsession. I get it. But we have a collective goal, and that's to engage beekeepers so we can learn and teach and start to be surrounded by very careful and educated and vigilant beekeepers, because that really matters to all of us. So tonight we'll discuss how we can cultivate a community of engaged and competent beekeepers. And I'll talk about educational programs, community, belonging, and leadership. I'll feature some of the things that have worked well at OSU, so what to watch out for and what we can keep in mind for the future events. And maybe if we have time, if we can figure out the speaking with the group, maybe we can share ideas so we can adopt them in our own areas. So let's start with the Master Beekeeper programs. There are Master Beekeeper programs in just about every state. They are time-tested, they're structured, long-term training, standardized and objective testing and evaluation. There's affiliations with universities, which connects people with science, clubs, and state organizations. It creates a pool of knowledgeable and reliable beekeepers, which is really important to spread that knowledge about bees and bees as pollinators to the general public and to beekeepers to make them better beekeepers. And of course, they build community. So the Oregon Master Beekeeper, this is a little on the structure of what we have here. We have the apprentice level, where we match a mentor with a student and they have hands-on training at that very beginning level. The journey level is all about community service, which is probably my favorite part of the journey level, self-paced learning, and we have field days with a little bit more advanced topics. The master level has includes outreach projects. They need to develop some kind of proficiency that's new to them. So that might be queen rearing or selling nukes to others. And it includes more science. So I'm going to show you a couple of master level projects that I really like. These are master beekeeper students assisting with an oxalic acid vaporization study. So these master level students got together and we all talked about the study, and they came and they're actually collecting data. So we have a real project for them. What they're doing in this picture is tracking larvae as before and after they were treated, and using this clear overlay so we can track them and see if they're alive or dead or what stage they're in. So projects like these, they allow the participants to get to know each other and learn together and explore new ideas. And what I really love is that beekeepers, they come from all different backgrounds. And so there might be a mechanical engineer in here, there might be a 5th grade math teacher in here. And so everyone's going to approach a problem in a different way. And it's really fun to hear them talk and explore new ideas. Another master level project that we had was this guy here in the picture is Paul Stromberg, and he had a project comparing the efficacy of different Varroa sampling methods. So he's telling us all about his protocol and the different ways of sampling for Varroa, and then we all helped him take all of the samples. And so through these master level projects, they're building competence, they're building confidence, and they'll take this knowledge home to their home region and share it with other people. So Paul, he's that type that probably could have done all of this without the Master Beekeeper program, but it's just a program that I think provides avenues and connections with other people. So it just kind of brings us together, and we can all be a part of it. He now teaches queen rearing classes in his region, and so I think it's really exciting to see how people move along through the program. We have, my favorite part of the Master Beekeeper program is all the hands-on training. This is a disease workshop. So I keep two freezers full of diseased frames. And my favorite thing is to bring them out and have people pick through them and smell the smells, look at all the different angles with the light, and really get to know diseases. And there's not always an opportunity for people to do that. So if you can do that with your club, have some kind of reserve of diseased frames or problematic frames that people can look at hands-on, it makes such a difference in learning. So hands-on learning is so important. And we also have a lot of time for fun. This is Bee Beard Day. It's not just about fun, it's about learning. So we're learning about pheromones through party tricks. Bee beards are really, they're fake swarms. And so we talk about pheromones, we talk about the swarming process. We put bees on all kinds of people. This guy on the left is the dean of OSU, of the College of Ag. And so, no, more than that, he's a higher level dean than that. So it was exciting to put bees on him. I realized I had been doing bee beards. I was trained by Dr. Mike Burgett, who is the predecessor of Ramesh Sagili, who's the current honeybee professor here at OSU. And so I had been doing this with him for 10 years. And I thought it might be kind of fun someday, since you guys are the real movers and shakers, the leaders of bee clubs in this. Well, I'm surprised to see how many people are from the West side, but Westerners too. So maybe someday we could do a little workshop where we can start a bee beard, the whole process from start to finish, and learn it as if we're putting on bee beards on people and not just being spectators or wearing them, although we could do that too. So just some more ideas. My favorite thing, we all have field days, of course, but my favorite thing is to have themed field days. So we've had a field day called All Hail the Queen, and we do everything related to the queens. We did hygienic testing. We got out all the pens and practiced queen marking using drones. People love doing that. We evaluate queen performance. We do some alternative queen rearing. We talk about pheromones. And then we also incorporate fun. So my favorite thing to do after hygienic testing is making liquid nitrogen ice cream. So there's a lot to this picture. It's about food. It's about community, friends, talking, and it's about science. So we're talking about how liquid nitrogen can freeze and make ice cream. Food brings people together. Having events that evolve around food, it makes people happy. They go together really well, and it gets people talking, warmed up, and ready to connect and build community. So make sure you have some kind of picnic or social event involved with your learning. We also go on DCA hunts. This is a really fun thing to do on a field day. We get these car dealership balloons and we tie pheromone or even a queen to the balloon and walk around the edges of these meadows. And while we're doing that, we're talking about pheromones and we're learning so much, talking with each other. So it's these ideas are they're just ways to incorporate fun and learning together. At OSU, I started Friday in the apiary, let's see here, back in 2014. I realized, well, we all realized there's a real need for hands-on experience. And I think I would get so many phone calls about distressed beekeepers. They don't know what's going on with their hive. Could I come take a look at them? And we don't have time to make house calls, but sure enough, I went to somebody's house, and on the way home, I thought, this is not sustainable. I can't keep going to people's houses and checking out their bees. So let's bring them here. So in 2014, we started a monthly Friday in the apiary group where we just brought everyone out to our apiary. It's very casual, and that was really important to me to keep the expectations low. It's not a formal class. It's just for fun. We're just gathering. I call it a bee happy hour. So we just go through the colonies and we talk about what's relevant for that particular month in bee management. We have a different topic, so a loose topic. Again, I don't want it to be formal. And if we have a topic, but somebody wants to talk about something else, we're all about it. very flexible. And it's year round. And I'll tell you about all the different things that we've done in the past. And hopefully there'll be ideas for you and your club, and maybe you can give me some ideas that you have. So the keys to success for Friday in the Apiary: it's free, it's regular somewhat. If we get too busy or if there's a weather issue, we'll cancel, but for the most part, it's pretty regular, and it's hands-on. And hands-on training is gold. There's just nothing like seeing a colony, especially for beginning beekeepers who have 1 or 2 hives. Seeing somebody else's hive, it makes a huge difference just to see differences. And of course, we know how different colonies can be in one apiary. So within Friday in the Apiary, we have these different little activities. So we have the Dead Beekeeper Society. We have a couple different ways that we've gathered as the Dead Beekeeper Society. First, this picture on the left, this is at the OSU library. And they have in their archives these incredible, it's an incredible collection of documents and books and photographs. Some books dating back to the 1700s. It's incredible. Sometimes we had to put on gloves to handle the materials. So it's a lot of fun to read, look at the pictures, look at the diagrams, and just gather together. And if you have a university around you that has had a bee program in the past, I'm sure that there are documents and old materials in the archives too. So librarians love it when you go up to them and ask if you can see something in the archives, because otherwise they're sitting in boxes collecting dust. So let's get these out and look at them and enjoy. It's amazing to see the really, really old materials, but then also this is a picture of a little bit more recent materials, like from the late 1800s and 1900s. We have a bunch of bee cultures and ABJs where the library didn't want them. There's just too much. Maybe they're a little bit damaged. And so I brought everybody together. We have a bar in Corvallis that's library themed. And so we all get together with food, of course, and we go through all the magazines, and we talk about how cheap the queens were or how glorious it must have been pre-Varroa. And then Bee Search, we also have a group where we gather together. It's hard to keep up with so much science in beekeeping and apiculture. And so we had everybody find a peer-reviewed article, and they had to come and give a little summary of what they found. So we were able to learn a lot about research in a social setting like that, and learn so much in a little bit of time. Friday in the Apiary, in the winter, we do things like fondant cook-offs. We have people come with their recipes. We cook fondant, we don't cook fondant, we have all these different ways. People, you know what it's like. Everyone has their own way. So in Western Oregon, we don't make a lot of honey, so we're constantly having to feed our colonies, and we have backyard beekeepers especially have to make emergency feed, so solid candy. So it's really fun to do that, especially around the holidays. And one time we even gathered a bunch of samples of everybody's recipes, and we sent them to the chemistry lab, and we tested for HMF, or hydroxymethylfurfural. And it was really neat to see the differences between cooked fondant, uncooked syrup, and so on. Like, if you add the acid, do you get more HMF, and so on. So really fun, and you can include some science in it. We also had craft night. So around the holidays, what better gift to somebody you love than a sugar shaker that is wrapped in glittered bows and stickers, and it has this instruction sticker right on it. So the beekeeper you love will be able to sample for mites with this beautiful gift that you make them. So we try to make it fun and social and learning at the same time. So that's all the indoor stuff. For the winter, it's sometimes hard to get creative in the indoors, but we do it, and we have a great time. So in the bees, We do general inspections, of course, everything's hands-on, and we have volunteer leaders to lead small groups. Most of them are Oregon Master Beekeeper participants, and they're earning service points to do this. This group here in the picture is a pretty large group because we had a spectacular speaker here talk about honey harvesting. But for the most part, we try to keep the group small so they can ask questions and get up really close. But we also keep things real. So when I started beekeeping, it felt like a lot of pressure going to bee club meetings because if I wasn't doing everything that they were talking about, I would feel a little anxious and I'm not doing it right. So we want to keep things real for people. So we have trouble in the apiary, and I try to keep, if we have a problem with a colony and I know an event's coming up, I'll keep that problem around just a little bit longer so people can learn from it. So trouble in the apiary, we've had a queen in the honey supers, laying worker colonies, bad queens. How do you know if they're bad? What do you do? And we work through all of it together. What do you do if you're not ready for winter? Let's not make people feel bad. Let's just figure it out and make it work. That's all about combining at that point. We have colony autopsy clinics. That's probably one of my favorite activities because you can learn so much from a dead out. It's been so great that we've created dead out detectives. So that's when we brought together higher level, more experienced beekeepers so they could learn how to have their own colony autopsy clinics in their region. And that's been really successful. It's a great learning opportunity, and it allows the beekeeper to learn and figure out what to do different next time. Okay, some things to watch out for. Watch out for open platforms. So you know the talkers and the storytellers and the advocates, and sometimes controversial issues come up, and they can derail your program. You can lose your audience that way. So you can people will start talking in the background, or just go away if there's something that's particularly offensive. Usually people don't talk about offensive things, but there's always, people that take up a little bit too much time. So just watch out for that and know when to kind of round people back together. Keep the content at the audience's level. So keep the beginning content for the beginners, advanced for advanced. If you do it as a big group, you'll lose people. So a great way to avoid losing people because of content is just to survey them before they come. So, or I suppose you could split people up into groups, but that doesn't work as well as splitting them up before they come. So ask the people that register for your events how much experience they have or what they want to learn, or so on. You may have volunteer leaders sharing misinformation. This is a hard one because so we try to have volunteer leaders at our Friday in the Apiary events and other Master Beekeeper events. We try to have them be people in the program. So we know they've gone to the classes. We know they've been trained through the program. But even then, sometimes you can get a wild card. So you just have to be careful. Do your very best. You know that you can't always. It's difficult, but you can try to kind of wrangle people back in and make sure that their information that they're sharing is credible. Watch out for talking too much when you're outside. So if you're talking, people are in their bee suits, like this picture right here. They look engaged and they're talking and learning, but if you're talking too much outside and people want to get in the bees, just go get into the bees and talk about the topic while you're there. It's going to be so much more effective. It's easy to get shifty when you're outside standing around too long. And don't make too much work for you. Don't burn out. If you're trying to make these events that are fun and learning and it's taking up too much time or too much effort, you'll burn out, and then everybody loses. You lose, and so do the people that need this training. So make sure it's not too much work for you. Maybe consider parting out some of the duties to other people so it doesn't all fall on you. And now for belonging. Beekeeping really is for everyone. Bees don't care who you are. All they care about is being managed, having enough food, being mite-free. So beekeeping really is for everyone. This is a picture of us, the Oregon Master Beekeeper Program in Spanish. We had our first year last year, and it was a great success. We had so much fun, and our second group is going to be this year. It's surprising how easy it is to find people that are bilingual and really eager to jump in and help. And it's amazing also to have a group of people where speaking in a native language allows people to relax, and then they can take in more information rather than just trying to puzzle together, translating in their heads. And so maybe some of these people in here do have they do speak English as well, but it's just having a native language and just that relaxation, it really makes a difference in learning. So it doesn't have to be a huge program. It can just be a bee day, some kind of bee event where you have a group that can gather together and speak in Spanish, or maybe you have a different language in your region where you can just round up somebody who can either interpret or translate. Other ways here's another picture of the beekeeping in Spanish field day. It's all hands-on, of course, and we have a lot of fun. More on belonging is accessibility. Consider having your apiary be on flat. Have some routes that are flat, that are stable for people that may have walking trouble, or this is a Valhalla hive that was developed by Naomi Price and her partner Larry, along with Vivian, who's here on the left. They made this long, it's a long hive with a hinged roof, kind of like a top bar hive, but it uses Langstroth frames. And I think this is so beautiful. This is one of my favorite colonies in our whole educational apiary because it's accessible for people in wheelchairs, and it's just for people who can't lift as much, or just maybe they have a bad back, or maybe, so it could be a disability, it could just be that they don't want to lift, but it's just another option. So accessibility is really important. And if there's a way that you can incorporate that in your apiary or in your events, it's just it's allowing more people to come in. So consider how you can widen your circles. When you have events, consider advertising in places that you normally wouldn't advertise. And you're just you're bringing in people that can offer different things and contribute in different ways. And it will make your events so much more beautiful. Okay, and kind of along with this is leadership. So leadership is, it's really hard for clubs to keep their leaders or have people sign up in a leadership position. So let's come up with some ideas on how to help get leadership roles. So start with small roles. Have people do small things. I put on here garbage duty. So my son just started playing baseball. And so I'm a new parent on the baseball team, and we had assigned chores to do for the baseball games. And so I got garbage duty because I am the bottom of the totem pole, and it sucks. It's a terrible job. I have to gather all the garbage and take it to the dumpster, and it's the worst job. But even when my garbage bag explodes and there's garbage all over the place, people come and they help me clean it up, and then I'm meeting them, and then I see them the next time, and then we talk and I learn their names, and then suddenly I feel a little bit more at ease in a group of people that I didn't know before. And so I feel like I'll probably get warmed up in these smaller roles and then want to take on more. And I think that can happen in a lot of different cases. So ask people personally if they'll do a role that they could fill, and just start integrating into the leadership roles. When people are asked personally, it's a lot harder for them to say no than if you asked a whole group of people and say, is anybody willing to take garbage duty? No one's going to raise their hand. Seek out the introverts. So this is a tricky one, because introverts, they don't like attention. They don't want to be asked out, but sometimes they're introspective. They have a lot to contribute. They know a lot. It just takes little steps for them to get more comfortable in big groups, but they are, if you give them some kind of a job, it really can make a difference. So giving someone a leadership role could change their life. And while you're not here to change someone's life, I've seen it time and time again in these kind of bee settings where they're just those real shy people, but suddenly they're learning more, they're becoming more comfortable, and they have a really amazing role and they're contributing so much to the bee club. So so seek out the introverts. Extroverts are great too, but it's not hard to get them involved. Extroverts are the leaders. We don't have to spend as much time trying to get their help, but introverts have a lot to contribute, so don't forget them. Okay. Bee club members, we need to keep the members coming. It's easy to get them here in March and April when their packages are here, their nukes are arriving, they're ready to learn, but August comes around and there's camping trips and rivers to float. So it's really hard to keep them at the meetings, but we need them because their healthy colonies mean everything to us. So how can we keep them? Food. So I've been to a lot of bee club meetings, and some are really engaging and fun, and some I just want to take a nap. One of the very best bee club meetings I've been to is they had a huge potluck. Every single meeting, they have this giant, gorgeous potluck, and they pull out all the stops to bring these dishes. And so before the meeting, people are eating and they're talking about the food, what's the recipe, what's in it. This particular place was coastal, and so they were all fishermen. They had this amazing seafood. It was incredible. And again, food makes people happy. They're happy to be there, and they're ready to learn. Maybe shorter presentations. A lot of bee clubs are on weekdays after people get home from work, and it's sometimes hard to stay through a long presentation. It's not lost on me that we are here at a long presentation, especially for those of you. I had no idea there were going to be some East Coasters here. So thank you for sticking around. I know how late it is. And I'm almost done with my presentation. So shorter presentations can make the meeting just move along a little faster. Okay, and engaging activities. Consider having some more hands-on activities in your bee clubs, group participation, and shared apiaries, kind of like Friday in the apiary. And prizes. I've seen some bee clubs have prizes. They do raffles. They get donations from bee supply stores, and I think that's kind of fun. And just, I don't know, I think just any way to get people engaged and feeling like they're part of the group while they're learning as well. You'll get them to come back. Some more ideas. Some of these ideas I've gleaned from newsletters of bee clubs around our state. They have frame building parties, which I think sounds really fun. There's a group that has bee buddy pods. So it's not exactly that they're mentoring, or it's just that they're just bunching beekeepers together, and they have bee babysitters. So if you're on vacation and it's time to treat for mites, you can call up your bee babysitter and ask them to just go slip in some treatment. I think it's such a great idea. It just makes people feel like they're part of something. It's more of a community, and you're helping. People love to help. Honey harvesting parties, of course, honey tasting events, bring out that honey wheel and do all of this together. Lots of fun. So so that's what I got. And this is my email address if you want to chat, but I think I'm going to stop sharing. And if there's a way that we could talk about what you guys do in your clubs, I think it'd be really neat to hear. If there's a way to do that, I don't know, but thank you.
Thank you, Carolyn. A lot of great ideas. I'll share you a couple of things that did come in from some others. Sherry from the San Joaquin Guild in California was emphasizing aspects that their club does with youth. with 4-H youth, with Grange youth, classes of one sort or another. You want to share anything that you feel from Oregon State that you can do more specifically with youth?
We don't do a lot with youth, and that's because with the Master Beekeeper program, we have a one-on-one mentoring component. And if we can't have kids involved without having to do a bunch of background checks. I think we could get a little bit stronger in that, but I think that our bee clubs around the state do a fantastic job at that. There's 4-H, there's the fair. I think there's a lot of work with youth that way. So yeah, that's just not our best, I think, at OSU. But we need to. They're our future for sure.
Yeah, I mean, a lot of the course, university students, which is the mandate for the universities. I mean, a lot with that group.
Oh yeah.
Jim Kleiman wrote in with a great idea for a summer meeting. How about a topic, dearth and drought? Are you dearth and winter? Are you ready? I thought that was a neat idea to get people to a meeting. And Liz had to sign off. She said, but a lot of food for thought in the comments of Carolyn. Another one, Joe Rawls is saying they've tried to budget 15 to 20 minutes for members to ask questions about their own hives and issues. This helps ensure that everyone gets a burning issue taken care of at the meeting. Other groups that seem to be very successful, is particularly for the newbies, is start the meeting a half an hour ahead of the meeting and have it be directed towards those questions of newbies. And then that is not done by one of the officers. We talk about burnout, but by a volunteer, one of the mentors within the group that is helping to handle that aspect, so that the officer, the president, can get about getting set to go for the meeting and things, and he or she is not tied up with this pre-meeting group as well.
Yeah, with the Master Beekeeper program, we do a monthly Zoom Q&A. And I had mixed feelings about that for a while, because it just felt to me like I was talking into a black box, and there's not a lot. And I wasn't sure how effective it was. But then I went to a bee school over the weekend, and people came up to me and said that how much they loved learning from other people's questions, and how important these Zoom Q&As are. Zoom has changed a lot of things, so we can actually we can learn so much from each other without having to go anywhere. We can eat our dinner while we're listening. And I was surprised at how effective that kind of learning was for them. So, so that's another way to do it for sure.
And Julie from Vancouver has talked about how important that open hive is. And in that type of a situation, tap other people to try to do that. This doesn't have to be the officers doing it all, doing everything. They can certainly parcel this out, and we've got as many willing volunteers. And it's a good way to keep some of the older people of clubs that know it all, to keep them engaged, because then they can teach what they know to others. We're going to do one other thing this evening, and of course we're going to have a live WAS conference. We're going back to a live WAS conference, and we have our director from the province of Alberta. We have settled for a date, so I'm going to ask Ron to come on and talk a little bit about what we're going to be doing and offering as our WAS program for a live meeting this summer, this coming summer, the end of the summer, in Calgary, Canada. Ron Miska is our representative from Calgary, from Alberta, and the meeting is going to be in Calgary. So Ron, are you ready to share a little bit about our meeting?
Yes, I am, just about anyway. I just wanted to add one thing to this excellent talk on ideas and engagement, and that is that here in Calgary, we have about 450 people in our Calgary Bee Club, and it's a very strong volunteer atmosphere here. And one thing that we do is any community event that we can showcase ourselves at, is somewhere to go with members' extra energy and desire to be in the public and to talk about bees. I'm talking like the Calgary Stampede. It's a huge event, a rodeo every year. We have Makers Fair. We have all kinds of farm market fairs and Aggie Days, and it just goes on and on. We have something almost every month that people can get engaged in. So that's just a possibility of something. But what I'd like to do is let's see. Let's move this out of the way. Okay, can you see what I see? Welcome to Calgary. Okay, thank you. All right, so I'm just going to go through this in 10 minutes or less, I hope. But I do want to just get some enthusiasm built up because we have a really exciting in-person conference coming up in the fall. And if you're not here, you're square. I mean, you just got to come up here. Calgary is a beautiful place. The conference is at a time of the year when you should be done with your extracting and have enough time to come up. It's just going to be great. We are going to have, let me see if I can move this ahead. I'm doing something wrong here. Sorry, folks. I'll get with it. Well, here we go. Okay. Why are we hosting in Calgary? Why not host in Calgary? We've got over 1 million people. We're a tourist city. We've got lots of stuff going on that's made for conferences and conventions. So we are excited to be able to talk about northernmost beekeeping, which is really different from a lot of other types of beekeeping that people throughout the West are going to be interested in. And the city is just made for this sort of thing. So we have a venue selected. It's called the Grey Eagle Resort. It's on an Aboriginal nation that borders on Calgary. This resort has been ranked as number 4 of all the hotels in Calgary. So it's a decent place to stay. It has good halls, and we can mix things up quite a bit for the different kinds of stuff that we're going to be showing. It's not going to intimidate you. It's not a really expensive place, even though it ranks number 4 in the city. It's quite casual and quite friendly. So this is where you want to come stay there. If you're all out of love, Air Supply is going to be performing while we're on September 30th. So you might want to get tickets for that because you're going to be staying and maybe sleeping a few feet away from the performers here. Anybody not know Air Supply? Okay, well, look them up. I mean, they are from a certain generation. They're very good. Okay, so where are we? We're in Calgary. Calgary is in Alberta, Canada. Not long ago, I flew to Chicago, and it was a $350 return fare from Calgary to Chicago, nonstop. I went to see my sister in San Diego. I think I paid less than $200 for that ticket, a return fare. Going into Texas or somewhere like that, you're going to pay a little bit more because it's one of those places that maybe is a little bit further, but also a lot of Calgarians do a lot of work in Texas. So it's almost, the flights are typically booked. You can fly down to Oregon for about $200 too. So we're not out of price range. The hotel itself is very reasonably priced, and we are going to be giving people plenty of opportunity to explore around our area. So the mountains are about 1 hour west of Calgary, and the Badlands are off to the east, just about an hour or so again, and day tours, day trips. Lots of fun and easy to do, and we're going to try to arrange as many of those as we possibly can for you. So why are we in Alberta? Well, we're focusing on what we're theming, the Northern Lights beekeeping. And Northern Lights beekeeping, then we do see the Northern Lights here from time to time. And under the Northern Lights, we are producing some of the most enormous and best quality honey crops anywhere in the world. We really are hard to beat. 200 pounds with normal beekeeping practices is quite regular for commercial beekeepers. The province of Alberta has an average over the last several years of 140 pounds per average per hive. And the reason that's a little bit lower than the 200 is that maybe 70 or 80,000 colonies of our bees are moved into canola pollination every year. And in canola pollination, those 70, 80,000 colonies of bees, which is about a third to a quarter of the colony count in our province, they don't make much honey at all. Sometimes they have to be fed during canola pollination, but we average that in. And we average in a couple thousand amateur hobby beekeepers who are also quite excited, by the way, to be up, that we're having this conference coming to town. So we have big crops, very long days. The bees are flying at 7:30, 8 o'clock in the morning, and they're going till 9 o'clock at night, and so is the beekeeper. Not flying, but working. The long winters, we're going to probably talk a little bit about that and the extremes in our weather here, and a lot more. The type of crops that we produce, the fact that canola honey is our main honey crop in Alberta, but it granulates very quickly, and how we have to handle that especially. So we are having a theme of Northern Lights beekeeping, but we're not exclusively restricting ourselves to just the north. So speakers are going to be coming with very general topics, very good new biology, experimental work, and as well as commercial beekeepers from our area who are going to be presenting for us too. So we do have a really good program set up for you. So it's a beautiful country, beautiful place to be. I mean, I love this picture. This is actually, I photographed this within Calgary city limits. So we've got 1.5 million people here, but we've got a lot of space to grow. And someday this will all be carpeted with a pavement. But if you come up this fall, you'll get to see this before it's covered. So I'm trying to urge everybody to get up here if you could possibly, please. So what's next? What do you need to do? You need to get up here. And we would like it to see people register as soon as they possibly can, because they'll get a bit of a price break by registering early. So you get that price break. Your rooms are about half price if you book them in the next couple of months instead of waiting until it's really close to the time of the conference. What we're going to have is a full day, our first day, our Friday of that weekend, which is the 29th of September. On that day, we are going to have workshops, and people will pay $50, and they'll be able to float around to all the workshops that are going on and learn about everything from disease inspection to 3D printing to writing your memoir about bees. I mean, we've just got lots of stuff planned for you. That last one, maybe not. Presentations will be on Saturday and Sunday. So that's going to go on for 2 days with our speakers, and we are still developing our speaking list. We've got some really interesting people, and some people who are really focused on the north and beekeeping here, and some who are on the cutting edge of some of the new developments in the bee world. So we're really happy about that. And finally, on the Monday, another optional thing that you could buy into, and that's to take a tour, a bus tour around to some of the beekeepers around the Calgary area, or else take off and head up into the mountains or off to the east and see our Badlands. But we also have some cultural themed tours involved too. So that's it. That kind of brings me to the end. I will post in the chat, I guess it's the best place to put it. I'll post a website that you'll want to go to. So let me do that. And if there are any questions or anything, I'd be glad to do that yet too. Okay, so let me figure out how to stop sharing my screen. And let's see, there we go. Okay, we're back. Lots of friendly faces again.
Okay, thank you. Thank you, Ron. Are there any questions for Ron? We have we only have a couple more minutes, but let's see if there are anything for Ron right off the top of the head. Anyone that has a question?
Okay, I see a question that popped up right now, and it's how many people typically attend the conference. It's typically between 100 and 300 people, and we have room for 300 folks to come in as guests and attendees. So we're hoping to push it up to that number. The price is very affordable, so I think we're probably going to make that. Yes.
Any others? Thank you. Thank you, Ron. Well, we have reached the end of our 2 hours. Appreciate your attendance very much this evening. We had a good attendance. I think some very excellent presentations. I really want to thank Dr. Delaney again for her presentation on diversity, and Carolyn for her discussion in terms of programming beyond university and within clubs to try to get members engaged and keep members engaged and keep a variety of programs. Appreciate it very much for both of you this evening. Many nice comments coming in for both of you. So again, thank you. We will have another mini conference next month, and we'll soon be getting information out on who that will be, the presenters, and a brief bio of those two presenters. So we'll see you here, same place next month. Thank you for joining us tonight. Thank you all.